Hello, class. This is the third recording in Module 12, the regulation of protein synthesis and protein breakdown, and it is the last slide deck that is specific to protein synthesis. In this recording, we are going to talk about mechanisms, intracellular signals that are well characterized at this point in the context of regulating protein synthesis. And as I mentioned in the second recording, these are highlighted steps. There are a multitude of other intracellular signals that have now been identified. I don't know if the recording is yet available, but yesterday Rutgers had an inaugural seminar by Dr. Don Lehman, who was instrumental in some of this work, specifically his graduate students, Dr. Tracy Anthony, Dr. Josh Anthony. So we're going to look at some of that pioneering data in this recording. And also, again, I want to make it clear, this is some of the work in my lab has been done with some of these signals in protein synthesis, as well as with regards to protein breakdown using human skeletal muscle, but it far exceeds my specific area of expertise. So I'm going to present this information to you from a practitioner's point of view and ask that you get a couple of major points from this particular slide series and recording. For some of you that are more well-versed or perhaps have the intellectual curiosity and have been working more towards molecular techniques and basic science, you can move further into the literature, and I would encourage you to do so on your own, to look at some of the newer material that is beginning to be forthcoming with regards to a multitude of other signals, their relationship to energy in the cell, and their ability to integrate the regulation of synthesis and breakdown. So with that preface, let's move on to talk about the mechanisms that we know are involved in regulating protein synthesis. First of all, we should all be comfortable with the critical role of amino acid availability to the body, and in the case of this discussion, to the muscle for supporting protein synthesis. I'm also going to return to the fact that individual amino acids have been shown to work as signaling nutrients, and first and foremost is the branch-chain amino acid, the essential amino acid leucine. Leucine can work independently outside of the mTOR pathway, and I'm confident that you've become familiar with mTOR with regards to insulin and insulin's ability to elicit different responses via the mTOR pathway specific to carbohydrates. But I think we have all gotten to the point now, as we wrap up the third and final macronutrient, protein, that the pathways are interrelated, and there's a lot of balance and integration that goes on between the utilization of fat, carbohydrate, and protein. So let's get back to mRNA translation and the fact that it is that initiation translation step that's the point where amino acids will mediate mRNA translation to stimulate protein synthesis. The asterisks at the bottom, specific to these initiation factors, particularly with regards to the eukaryotic initiation factor 4E, that is part 2 here, is regulated by leucine. And this is really where leucine is most highly regarded as a nutrient signal. In the context of regulation that we are talking about presently, the two steps that I'm going to ask you to appreciate and find yourself comfortable understanding is the dephosphorylation that advances initiation translation with the 43S pre-initiation complex. This needs to be done so that global protein synthesis can advance. And then also the fact that phosphorylation is key to regulating and making the 4E initiation factor available again so that protein synthesis can proceed. So in taking a closer look and in being conscious, all right, of not really moving these next two slides through animation so that we can talk about them in total, I have provided my notes for you at the bottom of each respective slide as we've moved forward that hopefully will help you when you go back to better understand these points. EIF2 here within the pre-initiation complex has a critical role as regulating the rate-limiting step of mRNA translation. In this step, EIF2 binds with GTP and the MET tRNA to transfer the MET tRNA to the 40S ribosomal unit. That GTP that's bound to EIF2 is hydrolyzed, and the formation of GDP and the EIF2 GDP complex is then released from the ribosome at the end of this initiation process. In that context, the exchange of GDP that's bound to EIF2 for GTP is required for this MET tRNA binding, and it's mediated by a second factor, this EIF2B. What's critical for you to note, and again, it's highlighted in the byline on the slide, is that dephosphorylation of the EIF2B binding protein is required to release the EIF2 GTP complex so that protein synthesis can then continue. This slide shows that second step where phosphorylation is required or activates protein synthesis by releasing, all right, the EIF4 initiation factor. This particular step is a little more straightforward when you look at how it is illustrated, because what you see is that the initiation factor, EIF4E, is bound to a binding protein similar to what we saw in step 1. These binding proteins are specific to the eukaryotic initiation factors, but what is clear in the figure is that phosphorylation of the binding protein is necessary to release the initiation factor so that initiation proceeds, eventually forming the 48S pre-initiation complex so that protein synthesis can then continue. Leucine has a role not only in the phosphorylation of 4EBP1 so that this initiation factor is released, but it also has a role in sort of upregulating the kinase that is specific to phosphorylating this binding protein, the S6 kinase. So I'm going to take you to the original paper that Dr. Anthony published as part of her doctoral work in characterizing the role of leucine with regards to stimulating protein synthesis in rats. And I am going to say I was very impressed to learn yesterday in remotely attending the meeting is that Dr. Anthony has continued her work with amino acids. I did mention her in terms of looking at amino acid sensing and focusing on adipose tissue and attempts to regulate different aspects of adipose tissue in the context of weight management. But the sidebar here is that she just received, as a PI with colleagues at Indiana University and the University of Minnesota, $2.6 million plus in grants from NIH to continue to pursue the role of amino acid sensing, branch chain amino acids in particular, but she has also tagged other essential amino acids in regulating various aspects of protein utilization for promoting health, preventing disease, managing disease, and extending the lifespan. So with that said, let's just take a look at a study that was done to look at orally dosing leucine with or without carbohydrate in rats, sampling the muscle tissue and looking at the status of the binding protein for EBP1, the release of the respective eukaryotic initiation factor, and rates of protein synthesis in an animal model. So there were two groups, a fed and a fasted group. And with regards to this food-deprived or fasted group, they either received saline or a carbohydrate meal alone or leucine or the combination of carbohydrate and leucine because of the role of insulin in signaling the mTOR pathway and the fact that the mTOR pathway is essential in the context of protein synthesis. With regards to insulin, we know that it stimulates amino acid uptake, it stimulates protein synthesis, and it may or may not affect protein breakdown. It certainly doesn't increase it. Some studies have indicated that it decreases. Others have said that protein breakdown doesn't change. But we would all agree that when protein synthesis increases and protein breakdown does not change or decreases, there will be a positive protein balance. So insulin in and of itself is anabolic with regards to protein utilization. So these methods, I'm not going to read them step by step. I'm going to defer to you to take time. But what you're going to see is that, in this case, an isotope of phenylalanine was used to look at protein synthesis. The initiation factors that they were interested in particular, was the EIF4E initiation factor, and they were investigating the effects of leucine on the kinase, which is responsible for phosphorylating the binding protein to release EIF4E so that protein synthesis theoretically would then advance. So this was some of the definitive and pioneering data that moved from animal models, and that still is the case for the very sophisticated studies that Dr. Anthony does at Rutgers. But also it was the basis for work that we did in my laboratory, where we really extended that or translated it to people, to human skeletal muscle, and then moving forward to diet and exercise interventions. So in looking at this slide, again, I'm going to try to move it a little bit to keep it in the window for recording. You'll be able to better view it when you can do the same thing, but I want to walk you through The data here that is specific to changes in plasma insulin, changes in leucine concentration, and then ultimately changes in protein synthesis in response to being fat, to being ad libitum fed. That's what we have with F. S is the saline, which was gavage feeding. They put it into a syringe and fed the animals and then sacrificed them. So S could be the fasted or basal state. CHO is carbohydrate alone, L is leucine alone, and again, carbohydrate and leucine together. So what you see with regards to insulin is that in response to feeding, insulin is higher than what you would note in the fasted state. You would also note that whether alone or in combination with leucine, there is an increase in plasma insulin. And with leucine, there is essentially no change compared to the fasted state. The concentration of leucine in response to the feeding protocols was such that it was tremendously increased for the gavage feeding of leucine alone, and it was higher for the carbohydrate leucine group compared to either the fed or the fasted or the carbohydrate alone. Of interest to our discussion is the fact that, again, in the fed state, you have higher rates of protein synthesis compared to saline only. Providing carbohydrate and increasing saline really didn't have a major effect on skeletal muscle protein synthesis. However, the provision of leucine alone or in combination with carbohydrate increased skeletal muscle protein synthesis. So this is where you would be able to note that leucine is working through a pathway independent of insulin. With regards to the binding protein for EBP1, what you see here is the results for the fed, fasted, or saline group, carbohydrate, leucine, and the combined group. This is looking at the association, the binding of the initiation factor with 4-EBP1. So what I want to point out and what I've bolded for you here is that there is lower association of the initiation factor with the binding protein for the fed and the leucine groups. What that means is that when we look at phosphorylation, is that the binding protein has been effectively phosphorylated through the kinase with feeding and to a good extent for the leucine and the carbohydrate and leucine groups. So the major point is that leucine can independently stimulate protein synthesis, and that increase in protein synthesis is related to phosphorylation of the binding protein so that the initiation factor EIF4E has been released for protein synthesis to advance. Now that was an animal model. Shortly thereafter, Fujita and colleagues in Rasmussen's lab did a study where they were looking at these factors in humans that were involved in a feeding study where they looked at mTOR, they considered phosphorylation of the 4-EBP binding protein, they looked at the kinase, and then they also considered an elongation factor. All right. This study was done in men, and they used stable isotope and molecular techniques. After a baseline muscle biopsy, the men consumed either a leucine-enriched carbohydrate mixture, which is the nutrition group, or they had nothing at all, which is the control group. And 1 hour later, there was another muscle biopsy. And for both biopsies and blood sampling collections, they did the analyses for mTOR, 4EBP1, the kinase, and the eukaryotic elongation factor, EEF2. This slide shows the results for these outcome measures before the baseline biopsy in white and after in black the information following nutritional support or consuming the nutrition beverage. And what you can see is with the carbohydrate and amino acid mixture, essential amino acids only, there is an increase in mTOR. All right, again, a response you would get with a rise in insulin. You also note, though, that with regards to the 4EBP1 and its phosphorylation, that it increases, and this is simultaneous with an increase in the kinase, which would indicate releasing the eukaryotic initiation factor, 4E, and therefore the additional piece of information here was the decrease in EEF2, which is an indicator, all right, of, excuse me, the release of the protein from the ribosome. So in general, what they found when they extended that to measures of protein synthesis via the incorporation of a stable isotope of phenylalanine into the muscle is that leucine did indeed stimulate muscle protein fractional synthetic rates. So the primary role of dephosphorylation in the pre-initiation complex to get to that first step in moving forward with the MET tRNA complex is a critical step. And the other step that we focused on in this particular discussion of regulation of protein synthesis is the role of phosphorylation of the 4EBP1 binding protein and the subsequent release of the EIF4 initiation factor so that protein synthesis can proceed.